So, as Mohammed had alluded to, I'll be talking on really the early discovery aspects of antibody drug discovery. So, what I have here is essentially the outline of my talk, have an introduction, and then discuss several aspects of antibodies. Let me just get a laser here. So, antibody modes of action, considerations for antibody target selection, the different types of antibody platforms and tools, platforms for generation of antibodies. Speak a little bit about different xenograft models, as well as kind of in conclusion to wrap up all the themes that I've addressed, have a few moments to talk about some of the FDA-approved antibodies. So what I'm going to talk about now is essentially generating antibodies in the context of cancer, although of course we all know that antibodies obviously have been demonstrated to work in other types of disease or pathological indications such as autoimmune disease. But in the context of cancer, usually the way that we think of an antibody is really a protein that can block the interaction between cell surface receptors and their cognate ligands. Antibodies can, be employed to treat cancer in one of many, many ways. One of which is to enhance a patient's immune response, to directly block cell growth factors, impacting the tumor vasculature, or which is becoming actually more attractive, is delivery mechanisms by employing either a toxin or a radioisotope. And as you probably are all aware, there are a number of FDA-approved antibody drugs for the treatment of breast cancer, non-Hodgkin's lymphoma, chronic lymphocytic leukemia, colorectal cancer, and non-small cell lung cancer. And I'll get into some of the specific examples later on in my talk. So a number of points to keep in mind or to consider when initiating an antibody program is, first of all, what is going to be the function of your antibody? Do you want to block a growth factor ligand receptor interaction, as an example? Affinity. What is the affinity threshold that you need to impact or modulate your target of interest? Epitope selectivity is also very, very important. Isotype of your antibody, IgG1, IgG2, IgG4, which I'll address. Tissue distribution of your target. Is it widespread? Do you have a high normal tissue expression, or is it selectively upregulated within a tumor microenvironment? And species cross-reactivity. Does your antibody, for example, cross-react to a mouse or a rodent ortholog? This is very important when you have to design preclinical mouse studies. If you have an antibody that does cross-react to the rodent, you have a tool that you can use to basically evaluate and profile your antibody in a mouse rodent system. So in terms of antibody modes of action, there are a number of points to consider, different mechanistic angles. One is, as I alluded to before, angiogenesis. Are you able to potentially target the tumor vasculature or block angiogenesis, essentially feeding the blood supply to a tumor?